Welcome to the Masters of Beautiful Achievements podcast, where we share the insights from inspiring masters about how the use of physics, biology, and green chemistry is shaping our future. It's all you need to know about innovation and better business models. The podcast is hosted by Alexander Prince, founder of Scope Matters. For more inspiring episodes, visit the Master of Beautiful Achievements website. Today's episode is part of a special series where we look at microbes through a different lens. We will explore how life sciences, biotechnology, and environmental sciences are influencing how we look at our natural world. During this series, Alexander Princeton and Mark van Loosdrecht are curious to know what happens when we zoom out and look at microbes as important players in a much broader system. How does this different perspective influences on how we look at the role of microbes and viruses in the web of life? And what lessons can we draw from nature's logic into making our world more sustainable? Together with specialists in the field of virology, microbiology, and biochemistry, we go on a journey combining insights from various disciplines to learn about the essential questions we might have been overlooking. And who knows, we might discover one or two innovative applications along the way. This series is brought to you by Scope Matters, a systemic innovation agency from Rotterdam, the Netherlands. They accelerate business leaders through systemic innovation by merging ecological thinking with economic doing. Their system approach transforms and improves the business model of their clients by creating new opportunities, mitigating risks, and increasing cash flows, while at the same time eliminating waste and doing good. It is the space where systemic sustainable thinking meets circular innovation, because in the end all matter counts. Check out the website on www.scopematters.com. And Orvion, a young biotech company based in Stolwijk, the Netherlands. They study microorganisms and are developing their own DNA analyzing techniques, like for instance the Orvion U-Detect. They work on solutions to remove contaminants from ground, water or industrial processes more cheaply and permanently. Their services are available for water treatment, soil remediation, ecology and industry. Orvion believes nature can do a much better job. So please take a look on their website on www.orvion.nl. So the title of this session is Microorganisms Through a Different Lens. I want to thank you for Venture Cafe for hosting us. Today, Mark van Loosdrecht, I have to specify this time, and myself are hosting this monthly session here at Venture Cafe because Mark and I are extremely interested in microorganisms and especially how we can look through them through a different lens and find unexplored angles to see what this small kingdom or big kingdom has to offer. It's a podcast series hosted by the Masters of Beautiful Achievements and by me, Alexander Princeton, and Mark van Loosdrecht. And during this series, we will look at our natural world from a different perspective, and hopefully we will discover how it impacts how we look at life sciences, biotechnology, and environmental sciences. And for each session, we will invite a guest speaker in a field of virology, microbiology, and chemistry to explore certain topics. And for today's session, we're gonna investigate the recent insights about bacterial biofilms, specifically what we can learn from the role of Sialic Acid, and what are the possibilities to use these new insights for industrial applications. And eventually, I assure you, we're going to learn a lot about microbes and how they work in these micro biofilms and what Sialic acid has to do with this. So my name is Alexander Princeton. I have a company which is called Scope Matters, and we help companies to accelerate their innovation capacity by merging ecological thinking with economic doing. And how we do that is to apply natural sciences with systems thinking to develop innovation roadmaps, which eventually will do better. Very concrete, we have dialogues and we implement them towards innovation programs, because eventually all matter counts. Today we have a guest speaker, Mark van Loosdrecht, a professor from Delft University of Technology. Welcome, Mark.
Yes, happy to be here.
And could you introduce yourself in a couple of alineas about who you are and what you do.
Okay, yes. Yeah, so I'm Mark van Loosdrecht. I work already since 1988 at Delft University, currently as a full professor in environmental engineering. I studied in Wageningen universities, environmental engineering, a mixture of chemistry and microbiology, and did also my PhD in Wageningen on the interface of microbiology and physical chemistry, on how bacteria adhere to surfaces. And when I moved after my PhD to Delft, I more or less continued that line of work by not studying how bacteria attach to surfaces, but how they grow on surface in biofilms. And in Delft, so I'm heading the group in environmental biotechnology, which is a quite extended group. But what we especially do is trying to understand, on one hand, microbial ecology, what drives microorganisms, and then design systems based on the understanding of microbial ecology in a way that where in the traditional biotechnology, you would say empty a reactor, clean it completely, put the organism in you want to have. Our approach the other way around. We create a process in which the organisms we want to have feel happy and will accumulate themselves into the system.
Okay, then we're gonna enjoy talking to you today, Mark.
Yep.
Yep. Then the other Mark.
Yeah, hi. I'm Mark van Loosdrecht. I'm a founder and owner of Orvion, a biotech company based in the Netherlands, near Gouda. We exist for almost 8 years now, and yeah, we work with microbes every day. We look at them from the water and our environmental perspective with the molecular biological techniques, using DNA and RNA to characterize them. And we look a lot at degradation of all kinds of compounds by the wondrous nature that microbes are. And yeah, I'm really interested to talk to Mark today about many aspects of his work. So nice to have you here, Mark. Yeah.
Yes, always pleasant about work.
Yeah. So I did some research, Mark.
Yep.
And this space is huge. So I have to kick up with a question, and then we just go on the roller coaster.
Yep.
What are biofilms and why do they matter?
Well, you have 1 hour or something.
Yes, 1 hour.
Yes. Now, biofilms are maybe more or less the natural way how bacteria grow. That's in nature. Almost everywhere bacteria grow on surfaces. They immobilize themselves on the surface, and they do so by creating a slime layer in which the cells are embedded. And the most well-known biofilm is probably the one you try to combat every day on your teeth. When you brush your teeth, you remove a biofilm, essentially. So tandplak is a biofilm. If you have your gutter where the water from your sink goes down, you feel a slimy layer, that's a biofilm. In a river, they are covered with a slime layer, which is a biofilm.
Could you then say, Mark, sorry, could you then say that biofilm is actually the soil of a bacteria?
No, the biofilms is the immobilization of bacteria. They immobilize themselves. So the bacteria which grow on your tooth, or your teeth, if they would not adhere to your tooth, they would wash away to your stomach and don't survive. By attaching to the surface, they immobilize themselves, and all the sugar you eat, they can consume for their growth. And that's the same in nature. Everywhere water flows. So if you're in the water, as a microbe, you've flown away with the water. Whereas if you attach to particles in the sediment of the river or on stones, then you stay where you are, and the food passes by, and you take the food which is coming in.
Yeah.
So it is the natural, for a large part, the natural way of behavior.
Yeah, it reminds me so much about mussels. seashells that actually attach themselves just to ensure that they don't flow away.
It's the same principle. You can consider maybe if people want, they can consider such a muscle field also a biofilm. It's a layer of biological material. It's a matter of definition, and the biofilm itself is not very well defined. Just it's a layer of bacteria in general, if we talk this microorganisms, but there might be also higher organisms, protozoa or small worms in it as well.
For those who are not biological experts, what is protozoas?
Oh, yeah, protozoa is another microorganism, but they are already about 10 to 20 times bigger than microbes.
Okay.
So from a microbe point of view, it's already a very big animal which eats them.
Okay. Mark.
Yeah, Mark. So what you actually say is that biofilm is probably just a way for microbes to stay where they want to be, where the food is.
From a first perspective, it's for them the way to put themselves at the spot in nature where they grow best and to stay there. And of course, they partly are sometimes mobile. So most cells, you have the biofilm stage and they have the planktonic stage.
Yeah.
Otherwise, not come at new places.
Exactly.
And that's how they distribute over Earth.
Yeah.
They when they are at a place where they are happy and want to stay, they immobilize themselves in a biofilm. And if things go bad or they lose some cells, and they are distributed in the environment until they reach a spot which is nice.
Because I recently read about Geobacter, this very special soil bacteria, that they are motile, so they can indeed they can also choose to go afloat in a way, and then get to the food and then probably adhere again.
Yes, if there's enough food, then they will adhere again, and
and of course there are bacteria that have flagelles as well, so that can swim and grow towards.
Yeah. Long distance is just with the flow.
Yeah. Go with the flow. Yeah.
Short distance is with the flagellum, but very tiny.
Then I have a question to you both, just out of my curiosity. I mean, if you look at the emergence of planet Earth, our spaceship, I mean, the talk is that eventually there were bacteria and then the higher species emerged. But the way you describe it, van Loosdrecht, is that the immobilization is sort of a way of how life will emerge. And I cannot pull my thoughts away from that idea.
Yeah, that's an interesting idea. Well, of course, it is how life emerged, because bacteria are the early life and they make biofilms. And it's sometimes compared to that biofilms are, say, the early stages of multicellular organisms, which I personally think is overstating, but you could think about it, because in the end, our body is not much more than a bunch of cells which are embedded in an extracellular matrix, and that's the same as for bacteria.
Mm-hmm.
And also in our case, our own cells make this polymers which embed then the cells in that polymer matrix.
Could you elaborate a little bit on that for those who find it one step too far?
Yeah, so in a biofilm bacteria, they are single cells, and they grow, and while they grow, they also produce some slime gel material. That can be all kind of polymers, but you can think about gelatin as the most common known ones. Alginate sometimes is used, carrageenan, but they produce these polymers and they embed them in that polymer, just like if you make a dessert with a gelatin pudding, where you put fruits in, you immobilize the fruits in the gelatin pudding. And that's what bacteria do then at their own scale. And in higher organisms, it's also cells living they're bound together by these polymers. And in that respect, it's similar, but of course, in a higher organism, it's really not the individual cell which functions, but it's the combination of cells which function. And in the biofilm, it's essentially the single cells which function, and it doesn't function in the same way as a tissue. despite a lot of talk about it.
Yeah, and what I find interesting about biofilms is I would like to know your opinion on that. But it's also true, I think, that bacteria create their own environment in a way within this biofilm, because the conditions can change because of when there's a thick layer of biofilm, it gets anaerobic inside, for instance. You get completely other microbes to grow there.
Yes. Yeah, here's a clear difference between biologists and engineers.
Ah, good.
Biologists will always say that bacteria create their own environment, and engineers will say bacteria make use of the environment which is there.
Ah, okay. So I'm more a biologist than an engineer.
I'm a bit a chicken egg issue, but more that bacteria are just opportunistic organisms, which if they find an environment where they can grow, they grow there. And if a biofilm grows thick, the inside becomes anaerobic, and then you get bacteria which do not need oxygen grow inside.
Exactly. I was thinking about microbial induced corrosion on steel pipes.
Van Bemmel, can you explain that a little bit more?
Yeah, if you have water flowing through an iron pipe, for instance, then on the inside of the iron pipe, you get a growth of bacteria which form this biofilm, a slime layer. And when it grows thicker and thicker, then inside the layer, the oxygen that's in the water cannot is used up in the first part of the biofilm. So in the deeper part, you get strictly anaerobic conditions, and which are nearer to the iron surface. And there you can grow, bacteria can grow, which are strictly anaerobic, which also damage the iron by producing sulfuric acid, for instance.
No, that is true. But essentially the bacteria which grow in the inside are not the ones who start making the biofilm.
No, exactly.
They in the end just make use of the fact that there is no oxygen. And so that's why, from an engineer point of view, it's making use of it instead of organizing it.
Yeah, but of course both are true, I think, in a way.
For designing and understanding how you have to manage the system, it's better to understand if the bacteria are having a kind of directive way of doing, or whether they are just opportunistic organisms, where if you provide them the right conditions, they will do the job for you. That's, at least from an engineering point of view, that works much better than the other way around.
Yeah, of course, you're certainly right, Mark.
And what I've researched is that biofilms, a biofilm is full of various polymers and acids and whatever it's in there. Could you elaborate a little bit on that?
Yes. So here is a bit of link now to what we are currently doing in the lab. So that idea of bacteria being of biofilms being a kind of predecessor for higher organisms. That's I think that's a bit over the top. That's people say, okay, in a biofilm, as Mark also just said, you have different kind of organisms, always their own functions. And like if you have a organ, you also different kind of cells, each with their own function, and one could evolve in the other. And likely that is somehow how it's go, but this is in my view a bit too far-fetched. But the interesting thing which we observed in recent years is that initially the idea was that slime layer was a kind of simple polymer. And like gelatin or like alginate, just a kind of polysugar in which the bacteria embed themselves. And some bacteria do that, but what we have seen in recent years is that a lot of bacteria make all kinds of polymers, which are more or less the same polymers as the polymers which bind our cells together.
No way.
So, in a sense, you could see the biofilm as a predecessor of multicellular life, but more from the extracellular matrix than from the combination of cells itself.
What is an extracellular matrix? So
the extracellular matrix are the polymers which bind all these cells together in the biofilm.
Okay.
So the easiest way to compare it a bit on macro scale is the gelatin pudding, where you put fruits in, and then the gelatin binds all these fruits together into the pudding. and that polymer is then the extracellular matrix in which the cells are hanging, or the fruit is hanging. And what we see, where initially this was the idea that it's just a kind of sugar coating with maybe some proteins in it, and we now see that there's all kind of polymers, like with difficult names, but they look similar to, for instance, collagen or sulfated aminoglycans or peptidoglycans, and they resemble very much the molecules which bind our cells together. So it might be that slime layer, that polymer material is the most evolutionary predecessor of the higher life forms, whereas the cells have then evolved later on in that matrix. And that gives a bit of different view.
And also, you maybe we can make the bridge now to your sialic acids, because what I read about it is that you have found that these sialic acids that were usually also only associated with pathogens, human pathogens, that you find them in many types of biofilms right now.
Yes. Yeah, this is one of them. So there's a range of compounds, and sialic acids are currently an interesting one. So sialic acids are on all our human cells. All on the outside there are sialic acids, and they're used for recognition. So if you have compounds in the body which has to go from one organ to the other, it can be recognized in which organ you are by the type of sialic acids on the outside of the cell. There are many different ones.
Wow.
And that's recognized, but they also protect the cells. And for instance, currently, the coronavirus enters our cells by recognizing the sialic acids on the epithelium, binding to the sialic acid, and then cut away the sialic acids from the epithelium cells, and then invade the epithelium cells and start growing there. And so they are very much partly protecting and also the recognition points for instance, for viruses, for good and for bad things. And it was known that bacteria which live in the gut have sialic acids, but there was always thought that they take the sialic acids from the human host and decorate their cells with these sialic acids, and then the what's in English, the afweersysteem, the defense system of our body, doesn't recognize them anymore, because they see the outside looks as the sialic acids of the cell, and they don't attack these bacteria. But
camouflage,
it's a kind of camouflage, yes. It's just hiding away for the immune system of the cell. But first, if you look on the genome, you can see that they have the enzymes to produce the sialic acids themselves. And if you then go and look around in all kind of other bacteria, you find effectively that all bacteria can make these sialic acids, which play a crucial role in our bodies for recognition, also for good things as well as for bad things, as I said with corona, but also cancer cells. The sialic acids are a very important role in cancer development. Bacteria already have these molecules, and that was a bit a surprise. That was also a surprise, because many, again, there was thought, if you read in evolutionary books, etc., that sialic acids were invented at the time of the sponges or something like that, at a bit higher organisms. And the sialic acid, for those who have a little bit chemistry, it is like a glucose molecule, but the glucose molecule has 6 carbon atoms, and this is a sugar with 9 carbon atoms. So it's 1.5 times bigger than a glucose molecule, but further it looks like a glucose molecule, a sugar molecule.
Wow, that's amazing.
And that's again, that's why it's interesting to see that all kind of these compounds, which originally were thought to be quite unique for higher organisms because they deal with cell recognition and other things, are already present in early evolution with bacteria. And most likely the main role at the bacteria is just the defense.
But then, I mean, I'm moving to life sciences with my thoughts, the way you're explaining it, and what I know about cancers, and eventually everything is related to pH levels.
Yeah, somehow. That's
and the pH level is triggered by certain acids, which make fluctuate that.
That's not related here.
Okay.
The main thing is the charge they have. So they have a very strong negative charge. They are strong acids. And it's the charge which repels animals. And the defense is that if you have there are many different, I compared with sugar, but if you have a C9 compound, you have a lot of different ways to have the same compound in different forms. So if you have to enter a cell, like the coronavirus does in our body, you have to cut away these sialic acids first, before otherwise you don't get in. So most viruses have sialidases to remove the sialic acids in order to go in. Now what bacteria do, likely, but this we haven't studied, but this is now an assumption, they just modulate the type of sialic acids. Because bacteria get also infected by viruses. And if they would only have one sialic acid, it's an easy trick for the virus, because once it knows
always work. Yeah, you always get in.
But, yes, but there are dozens of different sialic acids, and each one needs a different enzyme to be cut off. So, and a virus will never have all this. In this way, a virus will be specific for organisms with a specific sialic acid, but the organism can defend by just mutating a bit and getting a different sialic acid. Now, that's for us not that easy, but this is part of why likely also people are less sensitive. Well, some people are more or less sensitive towards this coronavirus, because if you have the receptor, TC2 receptor as it's called, more in your body, you're more sensitive to it than people who have less of that receptor, and that's genetic variation.
Wow.
Mark, if I understand correctly, the sialics that are made by bacteria, but also by the human cells in your guts, for instance.
Yeah.
And do they differ, or are they the same?
No, they're sometimes the same and sometimes different. The bacteria seems to have a much wider array of sialic acids than higher organisms. Higher organisms have a few, but they have, of course, much more defense systems to the hostile environment than a bacterium.
Yeah.
And since they also play a role, or probably play more a role, partly their role in defense, but also for all kind of cell recognition, so that that it's recognizable whether you're in the lungs or in the heart for transport systems in the body. And for cells, I'm not a cell physiologist, so I'm not but it's somehow intriguing to see that molecules which have that much importance suddenly appear in biofilms, and they were simply overlooked because they are rather complex to analyze. It's not a simple analytical thing.
So if you would sort of try to make a biofilm without the sialic acids, they wouldn't function, or how important are they? Yeah, for instance, if you want to remove biofilm, you can think, okay, a significant part of the biofilm is a polysugar. Mm-hmm. If you add an enzyme to cleave the polysugars, it will not work. If you first cleave the sialic acids off, then you can hydrolyze the sugars. So if you want to remove a biofilm, that's why a lot of enzyme treatments work so bad. Yeah, of course, like the what's the name again? Proteinase K. Yeah.
If you not first remove it's not that they don't have any effect, but if you would remove the sialic acids, the protease works much better than when it's only when you don't remove them.
So the sialics are very important for the biofilm.
They are kind of part of the protection of the biofilm. And, and they simply protect by being highly variable. So other organisms need a lot of different enzymes to attack everything, and that makes it complex.
Van Loosdrecht, are you then suggesting that for me, this sounds very much like the plastics we make. We put a lot of additives in there just to make sure that they can't be broken down, and the way you're describing it is that the microorganisms are so smart to put like a potato. They put a
Yep.
very thin layer on the outside just to protect everything inside, and you can only attack it when you peel it off.
Yes. Yes, a simple. Well, life had to invent it, otherwise life wouldn't work. But as soon as you get viruses and you don't have a defense system, you have a problem.
Then my mind starts to move in innovation. If you put Sialic acids on, I don't know, on surfaces like we humans use, you might have a extra protection layer.
Yes, I think that's you always have to remind that bacteria and cells, they are a few microns, and we operate at the meter level. So there's a few orders of magnitude in size between.
Yeah.
And things which work on the molecular scale work very well in that science. But there could be all kind of use of sialic acids, and one of them is currently if you could produce sialic acids and you take them, they will bind to the receptors of the coronavirus and make that they cannot bind anymore to your cells because the receptor is already bound. So you can use them potentially in cures, but since developing sialic acids because this would be a medicine, not a vaccine, that production will take much longer than producing a vaccine, bring it to the market, because the whole sialic acids production now, there's a little bit going on because of cancer research, but yeah, that would be completely new way of producing compounds.
Yeah, and then I also have I mean, I have a face mask from a supplier from Rotterdam, some samples of the name I won't mention, and their technology is a surface where bacteria and viruses cannot attach to. What it actually is, I don't know, and I know there are surfaces where viruses are, I don't know, they cannot either attach or they break, the membranes break. How is that related to this then, to this type of acid?
Yeah, so that depends very much on the type of cells. So if you have an E. coli cell,
yeah,
very weak organism effectively.
Okay.
So I really don't know what they do, but I can imagine. I cannot imagine that they do this for all bacteria. I can imagine that for E. coli or for a virus, even for viruses, they have different ways. So like if you make the surface a bit hydrophobic and a coronavirus comes on it, it will likely disintegrate because this protein capsule will degrade, will deform. But if you have a more hydrophilic, it will probably stay like it is. For bacteria, there are many different ways where people try to make coatings where bacteria don't adhere, and this is always a very good sales argument. Now, if you are in the medical sector, in the medical system, where you like if in surgery, and you want to be sure that on your equipment for 1 or 2 hours no bacteria adhere, that's fine. That material you can make it. But if you think about something which more than 1 day will keep bacteria free, bacteria find ways to get on it. And the mechanism, I think what you said, that you can add a polymer with quaternary ammonia compounds on them, and that will be to certainly gram-negative organisms quite devastating because their external cell membrane will destroy it. But if you have this polymer capsule around it, which most wild animals have, you will not be that they will not be harmed by it. So I cannot judge about the company. I know a lot of people who advertise materials which are claimed to be antibacterial, don't work. And if you look to that test, they only look in short-term test of 1 or 2 hours, and then it can work. So for medical purposes, this can be very helpful in surgery and other things. but for more environmental, that doesn't work. Let alone if you have a face mask, you breathe. And if you breathe also air, but also all kind of organic molecules.
Yeah.
And within no time, that face mask is covered by your organic molecules from your breathing. And bacteria don't see the original surface anymore, but they see the surface created with all the organic compounds in your breath.
Yeah, then they migrate to the mask. Yeah.
It is.
Of course.
It doesn't help that much. This kind of things. You can include quaternary ammonium compounds. You can include silver, which certainly will kill bacteria. It certainly will help, but not on the long term.
Yeah. Okay, I have another question for you, Mark. Do these Cialics also play a role in the DNA exchange between bacteria, for instance, with plasmids or?
No idea.
You don't know, right?
I haven't seen that, and it's also I wouldn't think so, but that's
It's your guess, yeah.
Guess, yes.
I was just thinking that because, of course, the viruses carry DNA or RNA, and of course, bacteria also have their ways to exchange DNA.
Yes.
So I was thinking maybe these Cialics could signal somehow if that's wanted or not wanted, for instance.
Yeah, so bacteria would have the capacity to recognize, of course, Cialics, and because likely they would be able to make recognition molecules, binding molecules. But I've no idea. That's I haven't given it a thought.
Okay, good.
So that, for viruses, they have to move in, and they just have to find their way through the cell wall.
Through the membrane, yeah.
And they have the tricks for that. And I'm not a corona specialist. I only know that they have a sialic acid receptor and a sialidase on their surface in order to be able to get into the cell.
Yeah.
I'm just digesting this, Mark van Loosdrecht. How profound this actually is on the way we look at bacteria. And I found a quote that actually the sialic acid is a sort of a special sugar which they actually use also as an energy source.
Of course, you can. It's a sugar, so you can use it as the people of organisms which grow on bacteria. They will consume it and will convert it. They grow on it.
And because we know that bacteria will use hydrogen. We know that bacteria can use electrons.
Yep.
We know that bacteria like the sugars we know. And suddenly there is a new acid nobody have seen before, which can be an energy source. So suddenly the bacteria seem to have a palette of feedstock they can use depending on which metabolic process they wanna move towards.
But from a biology point of view, I always say in a complex way, every redox reaction which generates energy can be used by bacteria. You will find bacteria which can do it. Or in simpler terms, everything you can burn can also be used by bacteria. And that's not completely true, because otherwise the plastics would go. But
they will go, but it takes a bit of time, Mark.
Yeah. Most compounds to figure out.
Yeah, for instance, we've looked into PFAS compounds, polyfluorinated alkanes, which were made as forever chemicals. And suddenly last year, Mr. Jeffy from Princeton found bacteria that do degrade the CF bond, which was considered almost sacred. But in fact, we are going to release next week some data that we found them in Holland as well.
Yes.
Bacteria, they are just indigenous in soil and under specific conditions, of course. But if you find those conditions, you find the bacteria as well. and you will be able to degrade this CF bond of PFAS, which is remarkable. Yeah,
Mark.
I haven't. We worked a bit on fluorinated antibiotics and other compounds. Yeah. They are around, but yeah, the fluor is not, as far as I know, not a standard, not a compound in biology normal.
No.
So in the past with the chlorinated compounds, it was not that strange because there are a lot of natural chlorinated compounds, which bacteria use in all kind of ways. But these fluorinated compounds, I had no idea, and I know that for say fluor part of the fluor medicines, etc., they are degradable, and for PFAS that should also be possible. But likely, I didn't give it a look in the chemistry behind it. It's
very interesting. It has to do with ammonia. Yeah. Okay.
Yeah, from Bemmel. I mean, that's interesting. The talk we had with him. I mean, he did the research on plastics in the water soil, and who knows that this type of acid is actually one of these reasons why they can actually degrade plastics.
No. That's
no?
I don't think so.
I've learned you cannot exclude anything until you have seen it.
You can exclude things which you can explain from a pure physical or chemical way. So in biology, you cannot exclude anything, but in the end, biology only works if physics and chemistry work.
True.
And, because in the end, biology very smart exploits physical and chemical processes in a very efficient way. And things which, if you know there's chemical problems, then usually biology can also not solve it. The only thing is they can do it way more efficient and do things. And where if you want to produce ammonium from air, you do this at a few hundred bars and a few hundred degrees, tens of bars and a few hundred degrees Celsius. And bacteria do it at room temperature in open air. They're just way more efficient in catalyzing processes.
Right.
But
what is then what is withholding us as humans then to use bacteria to produce the chemicals we are synthetically making?
Technically nothing.
Okay.
There's no limit. We can make most of these materials we could replace by all kind of materials produced by biology. It's an economic, first an economic point, because the prices will in general be higher. And it's then if you really are going to do that, it becomes also a competition with food point at a certain moment. Now for materials not that quick, for energy it will, but you will have the problems. Now you can, of course, that's you mentioned already, bacteria can use hydrogen, so you could use electricity to produce hydrogen, or you could even use electricity to directly let bacteria produce things, but the costs are quite high.
But the cost is because the current bioreactors are expensive? Or?
No, the cost is because it's in water. And water is not a nasty, is for us a very fine liquid. But in chemistry, it's a nasty liquid. It's viscous. It has a high heat capacity, etc. So it's to get your product out of the water is a problem. If you have a product in acetone, you evaporate the acetone and you have your product. But evaporating water is, well, it's possible, but it takes you way more. So it's downstream processing and getting the product out of it, and partly the concentration. But so for some compounds, like PHA, where Gert-Jan Overing talked about last time, that is not too far away from market. But that's only maybe 2 times more expensive than oil-based plastics. But then you still see that's not going easy, because for all kind of reasons. We're looking with this EPS, these polymers from because that's where we're looking at for materials. The polymers, how to quickly say that, if you treat wastewater you produce bacteria, because you treat wastewater by having bacteria growing on the organic carbon, which say on the waste. But they don't do that for free because they grow themselves. And then in the end, you have the bacteria as a waste product, which in the Netherlands we now incinerate. But
Really?
yes, but roughly 1/3 of that material is biopolymers.
Yeah.
And with these biopolymers, you can extract them and make materials out of them. The composite materials we have shown, there's a range of, well, certainly for the materials part, you would need another hour to go in depth. But we can replace that. And the advantage there is that compared to PHA is that the cost price is lower because we don't need the reactor to produce it. It's already produced at the wastewater treatment plant. And you produce, the material you produce is actually these polymers you cannot make out of oil. That's different. So you're not going to compete with polyethylene. You're competing actually with the polyacrylates and other things which you can make from oil. But if used as a gel material, you cannot find it. And the difference is that if you have oil-based polymers and you make composite materials, that is a mixture again of, say, sand and polymers, that in oil-based polymers, you can maximum have about 20% of clay, and that's it. And then if you try to put in more clay, it becomes brittle and it's not a plastic anymore. If you take the biopolymers, like even alginate or the ones we extract from wastewater sludge, you can go up to 80% or 90% clay and still have a material which looks like plastic and behaves like plastic and can be used as a plastic. Now, clay is very, very cheap. And you bind the clays with this polymer, and this polymer gives the flexibility and the clay the strength. And you have a new material which could replace composites, current composite materials. And so there are options, new materials, and we can make, but replacing everything, it's more thing than how we would have to make materials also made more durable. And not what we do now a lot, certainly with the plastics, is use them once and throw them away within half a year or something.
Yeah, because I have a conversation together with in this case also one of the workers of Geert-Jan is also Groningen, isn't it? Yeah.
Mm-hmm.
Yeah. With the PHA, and it is a challenge to discover whether do you want to have a long carbon cycle of materials, or do you want a short carbon cycle of materials?
Yeah.
And if plastic is made as a long cycle, and it ends I don't know where, and if you have a shorter carbon cycle, then carbon is actually a feedstock or a let's say a drop-in chemical. You pee it, you poo it, and then everybody wants it because you can reuse it again.
Yes and no, because it's partly so. I know this discussion, and that's partly true. So of course the reason that like PHA and also our plastics we make, they are biodegradable. But in most cases we apply plastics because they're non-biodegradable. We don't need to put in fungicides and other things to keep it going.
True. But it depends whether you want the packaging for the long term or you want the packaging for the short term.
Yes, and then for the short term, of course, you remake it. But then the problem is that if you use, for instance, the biodegradability, that producing the material is still a lot of energy. So if you cycle it around, it doesn't go freely around. You have to
put energy in it. Yeah.
Yeah, but energy will be for free, Mark. I mean, if you see, electricity is everywhere. So
that's true, because but that's because it's still made out of fossil fuels.
No, but if you look at the microbial fuel cells and how microbes exchange electrons somewhere from the plant, I mean, electrons are everywhere. It's a potential difference, and if we humans start to use all the potential, I mean, then electricity will will be almost for free because it is everywhere.
No, I think electricity cost will always be low, but whatever you do, you need raw resources, because if you make electrochemical cells, you need electrodes with special rare earth minerals, and as soon as you start using more and more, these get depleted. So it is trying to minimize the cycling combined with the short cycle.
But then I'm curious.
Actions, not just one.
But how do your bacteria work in using electrons, and how do they exchange it? Because they have the geleider, or what's that in English? The conductivity.
Yeah.
So they have the conductivity materials to transport the electrons.
Yes. So some bacteria have that, and some they are a bit more conductive than others. A lot of bacteria can exchange anyway with metals like iron electrons, and then you have electron carrier or humic acids. So the bacteria have on the cell membrane a system called electron transport chain, and normally they donate these to, say, oxygen, but they can also donate the electrons to an electrode, but then they need a conducting material. But they can also just to iron or to humic acids or whatever compound, and if that cycles around, you also can transport electrons, not only through then conductivity, but also through molecules. So bacteria are wired up in all kinds of ways. But in the end, the energy they produce comes from glucose or from organic carbon.
And that comes from solar energy eventually.
Yes, something like that. Yes. So the bacteria never make energy for free. Only the plant-like bacteria, which use the solar light, they make organic material out of the sun. But all other bacteria, they have some organic carbon. They convert into energy, and in that step, there's always a bit of inefficiency, and sometimes a bit, and sometimes more. But
yeah.
Because they also want to grow on it. That's
also making this EPS or extra polymer stuff. Of course it also costs energy, I would say, but apparently it's worth it.
Yes, no, that's because that's one of the interesting things, because we see essentially that if you have slower growing bacteria, that they make more EPS.
Ah.
That's why E. coli has this, say, sugar coat, very simple EPS. And the slower growing bacteria, they make more of this, and that's a bit counterintuitive on some ways, because you think they have, that's like bacteria which have limited energy, they already grow slow, and then they go invest a lot of their resources.
In extra polymer substances,
polymers which they throw out of their cells.
I would. I mean, what is schildklier in English?
Thymus?
Yeah. That could be. Yeah, I mean, you have people with a very fast thymus and fast schildklier, and they are very thin, and you have those who are much slower, and they tend to widen up a little bit, and that's the metaphor I'm sort of jumping up in my mind.
Yes. No, that could be. But essentially, you can think out of it also in a different evolutionary way. E. coli grows extremely fast. So if some of the E. coli are eaten up by these protozoa I mentioned in the beginning, it doesn't matter.
Doesn't matter. Yeah.
But if you are a slow-growing organism and some members of your community are eaten up, you have a problem.
Yeah. Your life is more worthwhile.
More worthwhile if you grow slow, because if you are your longer periods before you divide, and so
that's interesting, Mark. If you're a slow grower, you have to defend yourself, of course.
Yeah, the rabbits have to, as a population, have to defend themselves less than tortoise or something like that.
Would you also say that the EPS producing guys, or the ones that you make a lot of this EPS, the slower growing organism, that they do metabolically more interesting things or more specific
yes,
things?
in general, because like E. coli is just fast growing.
Yeah.
Many of the others, they do special tricks.
Exactly. I was talking about this. I was talking about this acidic microbium PFAS degraders, for instance, but also dechlorinating organisms, the ones that are living under extreme conditions or doing extreme degradation processes.
Yeah. No, they grow on difficult. They don't grow on sugar cakes, but on very difficult substrates.
Exactly. And you often
like.
We do a lot of we do a lot of NGS work on populations under these very highly contaminated toxic conditions, and then you often see only a few species, very specialized, slow-growing organisms, anaerobics.
That's indeed.
Yeah.
So then, in that sense, it's logical that they protect themselves better and have to make it thicker coat to keep the environment out than E. coli, which divides every hour, and probably PFAS organisms, they will also divide just every day or 2 days, and I guess
maybe even 5 days, I think.
Oh, even 5 days, at least
way
slower than once per hour. That's
slow growers. But especially these slow growing organisms are interesting and do interesting stuff.
Just for us to slowly wrap up, Mark van Bemmel, just think about another question. I'll ask the audience, guys, if you have a question, please drop it in the chat box so we can pick it up and throw it at both Mark 1 and Mark 2, and see what comes out of there. For myself, van Loosdrecht, this is interesting. I mean, I'm also interested in quantum biology, and I mean, once you go into quantum mechanics and look at biology, a lot of stuff starts to make sense eventually if you understand how that works on such a level. But then you need to understand how the different acids and everything works as a whole system together. It used to be esoteric, and the guys who are working with flip-flops have an idea, but that's not quantified. But then now they're starting to quantify things on that nano level, and it's interesting to see that we're starting to discover so much more than we thought there would be. And that is sort of what I'm sort of confirming and learning at the both time.
Yes. I also see a question from Satoshi for Mark.
Yes.
If the coronavirus can recognize and bind to the specific type of Sialic acids, can we synthesize it and use it to absorb and remove the virus from water?
Yeah, in theory that's possible. That's for effectively it's affinity chromatography. The technique is more or less known. You could then you can take the receptor that is the virus bind, and in this way filter it out of the water.
Yeah.
Whether it's easier to add a little bit of soap, and they are also kit that. That's so. But it is a way to get them out and to concentrate them if that's of use.
But you would have to synthesize a very specific type of this Sialic acid.
Yes, the one they bind to.
Yeah.
Otherwise, you take the other ones also.
But for affinity chromatography, you effectively do that for recognizing cells or other things.
Yeah.
You just put then with antibodies usually, but you can do the same with lectins. Also, these are used, and it comes close to lectin-based chromatography.
Wow. Okay.
Yeah.
Then we're going to sign off. Both Marks, can you just stay on? And thank you everybody for joining this time. Next month we have another session. We'll announce the speaker a week before, and then hopefully we see some of you back again.
Yeah.
Thank you so much.
Thank you.
You're welcome.
I hope you enjoyed this episode of the Master Beautiful Achievements podcast. A special thanks to our sponsors Scope Matters and Orvion for making this series possible. If you like our podcast, subscribe in your favorite podcast app. And if you want to know more, do check out the mastersofbeautifulachievements.com website. If you like this podcast, do not forget to leave us a review on your preferred podcast platform. Thank you for listening and join us next time.